Molecular basis of inheritance need previous year questions from 16 to 23. Question 1. Unequivocal proof that DNA is the genetic material was first proposed by. The unequivocal proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase. So option B, Alfred Hershey and Martha Chase is the correct answer. What is the role of RNA polymerase III in the process of transcription in eukaryotes? In eukaryotes, there are three major types of RNA polymerases. RNA polymerase I transcribes 5.8S, 18S, 28S rRNAs. RNA polymerase II transcribes hnRNAs, precursor of mRNA. RNA polymerase III transcribes tRNAs, saRNA, 5S rRNA, and snRNA. So option B, transcription of tRNA, 5S rRNA, and snRNA is the correct answer. Expressed sequence tags refers to all the genes that are expressed as RNA are referred to as expressed sequence tags. So option A, all genes that are expressed as RNA is the correct answer. How many different proteins does the ribosome consist of? 80, 60, 40, 20. The ribosome consists of structural RNAs and about 80 different proteins. So option A, 80. Statement 1. In prokaryotes, the positively charged DNA is held with some negatively charged proteins in a region called the nucleoid. Statement 2. In eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. In prokaryotes, the negatively charged DNA is held with some positively charged protein in a region termed as nucleoid. Statement 1 is incorrect. In eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called the nucleosome. Statement 2 is true. Option D, statement 1 is incorrect, but statement 2 is true is the right answer. Given below are two statements. Statement 1, a protein is imagined as line. The left end represents by first amino acid C-terminal and the right end is represented by last amino acid N-terminal. Statement 2, adult human hemoglobin consists of 4 subunits, 2 subunits of alpha type and 2 subunits of beta type. In the light above statement, choose the correct answer. The correct answer is option D. As a protein is imagined as a line, the left end represented by the first amino acid and the right end is represented by the last amino acid. The first amino acid is called the N-terminal. The last amino acid is called the C-terminal amino acid. So option D, statement 1 is false, but statement 2 is true. Given below are two statements. Statement 1, RNA mutates at a faster rate. Statement 2, virus having RNA genome and shorter lifespan mutate and evolve faster. In the light above statement, choose the correct answer. RNA being unstable mutate at a faster rate. Consequently, virus having RNA genome and having shorter lifespan mutate and evolve faster. So option A, both statement 1 and 2 are true. The process of translation of mRNA to protein begins as soon as the largest subunit of ribosome encounters mRNA. Both the subunits join together to bind with mRNA. The tRNA is activated and the largest subunit of ribosome encounters mRNA. The small subunit of ribosome encounters mRNA. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNA and about 80 different proteins. In its active state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. So option D, the small subunit of ribosome encounters mRNA is the right answer. If the length of DNA molecule is 1.1 m, what will be the approximate number of base pairs? The two chains of DNA are coiled in a right-handed helical fashion. The pitch of helix is 3.4 nm and has a diameter of 2 nm. In the B form of DNA, the distance between a BP in a helix is approximately 0.34 nm, which means that there are about 10 nucleotides present in a complete turn. The total length of DNA molecule in nm is 1.1 × 10⁹ nm. The approximate number of base pairs 1.1 × 10⁹ divided by 0.34 is equal to 3.3 × 10⁹. 3.3 × 10⁹ is the right answer. DNA fingerprinting involves identifying difference in some specifics in the DNA sequence called as single nucleotides, polymorphic DNA, satellite DNA, repetitive DNA. DNA fingerprinting involves identifying difference in some specific regions in DNA sequence called as repetitive DNA, because in this sequence a small stretch of DNA is repeated many times. Option D, repetitive DNA is the correct answer. Identify the correct statement. The coding strand in a transcription unit is copied to an mRNA. Split gene arrangement is characteristic of prokaryotes. In capping, methylguanosine triphosphate is added to the 3' end of hnRNA. RNA polymerase binds with Rho factor to terminate the process of transcription in bacteria. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initial factor sigma and termination factor Rho to initiate and terminate the transcription respectively. Option D, RNA polymerase binds with the Rho factor to terminate the process is the right answer. Which is the only enzyme that has capability to catalyze initiation, elongation, and termination in the process of transcription in prokaryotes? DNA ligase, DNase, DNA-dependent DNA polymerase, DNA-dependent RNA polymerase. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all RNA. Option D, DNA-dependent RNA polymerase is right answer. Name the enzyme that facilitates opening of DNA helix during transcription. DNA polymerase, RNA polymerase, DNA ligase, DNA helicase. RNA polymerase holoenzyme binds to the promoter, unwinds DNA complex, and forms phosphodiester links between the initiating nucleotides. DNA polymerase, DNA ligase, and DNA helicase are involved in the process of replication and not transcription. Option B, RNA polymerase.